So this is lecture 15. In the last class, we've been talking about membrane transport and the importance of membrane transport. And we had looked at different types of carriers. So for a uniporter, it only moves a single solute. If we're looking at transporters that move two solutes, we can have two different types. With a symporter, both solutes are moving in the same direction. And with an antiporter, the solutes are moving in opposite directions. As an example of a uniporter, we looked at a glucose transporter. Remember, there are multiple glucose transporters. I think there's 15 in humans. We actually look at 2 of them. In this particular one, it's facilitated diffusion because the glucose is moving down a concentration gradient. And we can see how it binds to the carrier everts, and the glucose moves across the membrane. Channel proteins can also be used for facilitated diffusion, but in this case what we're doing is simply opening up a pore or a hole in the membrane that allows the solute to move through. They can be very specific, so just because you're opening a channel doesn't mean that it's non-specific. This example we're looking at here, the potassium channel, is actually very specific to potassium. Some amount of sodium or other ions may move through, but the specificity is quite high, which is a bit surprising. And we won't go into the details of why that is in this course, but you'll see it if you take Biochem 300A. So facilitated diffusion, we're moving a solute down a gradient, which is energetically favorable. So we don't have to input energy to do these processes. In contrast, for active transport, we're moving a solute up a concentration gradient, and we know that's not favorable. So what we have to do is we have to input energy in some form in order to carry this out. And two ways we do this is either directly linking them to a chemical reaction or indirect. And with indirect, we're using existing gradients. So in this example, we can see we're using ATP to drive protons, H plus, up a concentration gradient. So we're hydrolyzing ATP to ADP and orthophosphate, and we are moving a proton up a gradient, in this case, from inside the cell to outside. In the second one, what we're doing is we're utilizing that existing gradient, the proton gradient. It's favorable for the protons to flow back into the cell, and as they do that, they bring S, the solute, in, and the solute S here is actually moving up a concentration gradient, which would be unfavorable on its own, but because we're linking it to the favorable movement of protons, it will still occur. And so that really is the trick with the indirect active transport. So what we have in this situation is one of the solutes always has to be going down a concentration gradient. So let's say in our examples here that solute A is at a high concentration at the top in the blue area, probably outside the cell, and low inside. And in both of these examples, it's very favorable for A to move into the cell. What we can do is if B is going along for the ride, in this example on the left, what can happen is B, this is a symporter, so B is going to move in with A, and that might actually be up a concentration gradient. So it can be used for active transport. In an antiporter, again, our favorable movement of A in can be linked to moving B out, and that may be up a concentration gradient. And if B is moving up a concentration gradient, that's going to be active transport. The example we looked at for this was the sodium glucose symporter. We recall that sodium is at a much higher concentration outside the cell than in. So it's very favorable for sodium to move into a cell. As it moves into the cell, using this transporter, it pulls glucose along with it. And it can even pull glucose up a concentration gradient in this case. Okay? So if the concentration of glucose is higher in the cell than outside, this transporter can still be used to bring it through. because it's so favorable for sodium to move into the cell. After this, the cell will work to push the sodium out, and that may include things like the sodium-potassium exchanger we looked at that uses ATP hydrolysis to push that sodium out in a second step. But that's isolated from this transporter here. And the last thing we started talking about was looking at organelles. And one of the ways that we can study organelles is to purify or enrich them utilizing different centrifugation techniques. And you'll be looking at these directly in the lab. In the first one, what we're looking at is the rate at which something falls out of solution into the precipitate, okay, with things with a high sedimentation coefficient coming out first, and that's based on shape, density, size, that sort of thing. And in the bottom, we're looking at an equilibrium centrifugation where our band of interest is of a specific density, and the centrifuge tube itself has a density gradient in it, meaning that at the bottom here, the actual solute or solution is at a high density, and that it gets lower as we move up the gradient. We apply our entire sample to the top of the gradient and we start spinning, and what happens is that our organelle of interest will migrate until the density surrounding it is equivalent to its density, and that's called the buoyant density, and at that point it will no longer move through our density gradient. So we won't be doing that. It's kind of it needs some fairly specialized equipment as well as technical skills to do good density or good equilibrium centrifugation. So today we're going to talk about the endosymbiont theory. We're going to then look at the nucleus and the chromatin, which is how the DNA is packaged in the nucleus. And we're going to talk about nuclear transport and monomeric GTPases, and that will probably take up, I think, most of the time. If we have additional time at the end, we'll explore the endoplasmic reticulum. So just a couple of notes. Don't forget, I'll have office hours tomorrow from 10 a.m. to noon. Just use the Zoom lecture link in order to get in. And if you can't make it at that time, send me an email and we'll arrange an appointment, which will be sometime next week. Okay, is there any questions? Yes?
Last week when we were talking about the different types of centrifugation,
Yes.
you mentioned that one is more of an enrichment process than the other.
Right.
Could you please specify what that means?
So enrichment means it's not purified. So if you enrich something, it means that you are increasing the amount of it compared to other things, but that it's not pure. If something is purified, that means you only have that one component. And these steps are more, especially the velocity centrifugation is more of an enrichment process.
Thank you.
You're welcome. Okay. So again, not just plasma membrane, but really what we need to talk about here is the importance of membranes. So the membranes are what surrounds the organelles, and that's how we define and demarcate or limit our organelles. And that means that they'll have a unique lipid composition. So the lipid composition of the organelles will be different than the plasma membrane. They'll have their own complement of transporters and their own function. So there's a few different theories on different types of organelles, and different organelles are probably distinct. What we're looking at the left is an ancient eukaryotic organism, okay, so maybe a bacterium. And when we look at the bacterium, it's surrounded by a plasma membrane, okay, so that's shown in this gray line. And inside this gray material, we call that the cytosol. And within there, we have the DNA. We have some membrane-bound ribosomes. We'll see the importance of this in Friday's lecture, but the DNA is often loosely associated with the plasma membrane. It's thought that the way the nucleus evolved was via an invagination of that membrane. So if you can imagine you have a balloon, okay, and the DNA is associated with the inner surface of the rubber balloon skin, if you pushed your hand into the balloon, you could surround that genetic material that's associated with the inner membrane and enclose it. Okay, can you imagine pushing your hand into a balloon and then enclosing the DNA? And that's the theory as to how the nucleus arose. And if we had that, what we would expect surrounding the DNA, we would expect there to be two membranes. Okay? And indeed, that's what we end up with. So keep in mind this gray line around here is a lipid bilayer. And there's two of those around this. And so what we ended up with was this kind of unique compartment that's surrounded by two lipid bilayers. It also goes on, that process also gave rise to what's called the endoplasmic reticulum, which is the next organelle we'll look at. But because of this, the way it pushed in, it has a double membrane around the nuclear material. And so after this point, it's a true nucleus or eukaryote. So at this point, we have our first eukaryotic organism. And in this particular state, it's not a highly efficient organism. So it's not very good at producing ATP, the energy currency, in this state. So it is a eukaryote, but it's not the way you would picture a modern eukaryote. Okay? This moves us to what's called the endosymbiont theory. And one of the main proponents and developers of this was Dr. Lynn Margulis. Picture of her here. She died a few years ago. This many people have heard of. Yes?
What's the advantage of having a second membrane around the nucleus? Because I know for them, when we talk about like endosymbiont theory for the mitochondria, like it was an advantage for the cell to have mitochondria instead of other cells.
So the significance of the second membrane isn't necessarily that it's an advantage, but that's how it would form. So if we go back to our balloon theory, okay, picture you're pushing your hand into the balloon, okay? Everybody imagine this. Close your eyes, off to dreamland. Okay, just picture your thumb part of it, okay? So your thumb is pushing into that balloon. You're gonna have balloon skin on both sides of your thumb, right? And that pushes in, and it grabs hold of your genetic material, which we'll do with this thing of batteries, okay? You're with me so far? And then it closes up. It's surrounded by the rest of it. So how many membranes are around that? It ends up just the way it was forming caused two membranes, because you have the membrane was on both sides of your thumb, and when it closes up and seals, you'll have an inner membrane there and an outer membrane there. Okay? So the theory is that the reason it has a double membrane is that's how it formed. The additional reason that it's helpful is that it gave rise to this compartment here called the endoplasmic reticulum. And we'll talk about the importance of that double membrane for that in a little bit. And so we say that the nucleus and the endoplasmic reticulum, because they are derived from the same membrane, we call that contiguous. And that's a term we'll come back to, so we don't need to worry about it here. We'll see it in our ER part. Likewise, what we'll see is why there's a double membrane around the mitochondria in a minute. So the mitochondria, these are the powerhouses, the little factories making ATP in our cells. And Eukaryotic cells have two major factories. They have chloroplasts and they have mitochondria, and both of those are thought to be ancient trapped bacteria. So the eukaryote itself was never really any good at making energy. It only became good at making energy when it trapped bacteria. So the idea here is that what's pushing into the cell in this case is not your hand surrounding the DNA, but instead it's a bacterium. And the bacterium pushed into the cytosol of the ancient eukaryote. And the bacterium is thought to be similar to one called Rickettsia prausecki. Don't have to worry too much about the name, but that's actually a parasite that lives inside cells. And so it's possible that this started out as an infection where the bacterium entered into the cell, and lots of bacteria do this. Okay? So a lot of parasitic or pathogenic bacteria enter into cells. And so it entered into the cell, and what happened was that with time, they just came to benefit each other. And what the bacterium gets is protection, perhaps, and also the eukaryote can harvest nutrients that it may require. And what the eukaryote got in return was production of lots of ATP. And so with time, what's happened is it's become a single organism. And you can't take a mitochondria out of a eukaryote. If you take all the mitochondria out of an animal cell, the eukaryotic component dies and the mitochondria component dies. However, if you look inside a mitochondria, it has its own little genome. It carries out DNA replication, transcription, and translation. And those processes are all pretty much the same as what you find in bacteria, but they're different than what you see in the cytosol of the nucleus and the cytosol of the eukaryotic component. So they still retain many hallmarks of the bacterial cell. They're no longer separable, however. The mitochondria has lost most of its genes, and the functional components of the mitochondria, many of those genes are now actually encoded in the chromosome itself inside the nucleus. So it's this relationship which has been going on for a very long time. One more note I want to make here is when we're looking at a cell, everything inside the plasma membrane is the cytoplasm. So everything inside, and that includes the organelles. Okay? So that'd be everything here would be the cytoplasm. In contrast, cytosol, it's everything inside excludes the organelles. And so that's why we use the two terms. Sometimes it means we can use them more or less together, and often that's what we do. I'll try to use cytosol more often than cytoplasm, but sometimes I slip into using the term cytoplasm. So is there questions about that? So this turns out to be rather important because if we look at this particular slide, and let's go back here, what we can see is this area, change my color scheme here, this area here outside the cell, the extracellular fluid, that's trapped in here, isn't it? As this invaginated. And that means actually this area, shown in white, is similar to the extracellular environment. Likewise, this little area trapped in here around the mitochondria is also similar to the extracellular environment. We can call these topologies, and this is an oversimplification in many ways, but what we can see in this chart is we're looking at several of the organelles, and what we can see is that for ones that we'll talk about soon called, that are part of the endolysosomal system, we see that they're all colored pink. So, change the color up here. So if we look at the inside of the endoplasmic reticulum, the Golgi, endosomes, lysosomes, they're actually more similar to the outside of the cell than the cytosol. It turns out that the chemical environment of the cytosol, shown in gray, and the nucleus are the same. Okay. So the cytosol and the nucleus have the same chemical composition, but that differs from what we see inside the organelles. And for these particular organelles, which are part of this endolysosomal system we'll talk about soon, the inside of them is similar to the outside of the cell. And that's going to turn out to be very important for the cellular functioning of many proteins. And that we'll return to in a little while. So, just to give you an idea, these are generalizations, but if you're looking at the percentage volume of just sort of an average, this is looking at a liver cell. So most of it is cytosol, okay, that's most of the volume, followed by mitochondria, and then working our way down. This is maybe a little bit more interesting in some ways because what we're looking at is cell membrane here. And here we're looking at two different types of cells, a liver hepatocyte and a pancreatic exocrine cell. So liver hepatocytes, these are used for regulating glucose, blood glucose levels, detoxification. Those are sort of major steps that are used there. So that requires a lot of smooth endoplasmic reticulum, and we see that reflected here. About 16% of the cell membrane is smooth ER. In contrast with a pancreatic exocrine cell, they're involved in secreting proteins. Okay? And so that requires a lot of rough endoplasmic reticulum, and we can see that reflected here as well, with 60% of the membrane being rough endoplasmic reticulum. So again, we can make generalizations, say this percentage is this and this percentage is that, but it's different for different cells because cells have unique function. All right, questions just about the background there? No? Great. So let's start talking about the nucleus, and we have a couple neato concepts that are going to come up with this today. So the nucleus in a eukaryote contains almost all of the DNA. Where's the rest of the DNA? Yeah, in our mitochondria, or if it's a plant, it would be in the mitochondria as well as the chloroplasts. We didn't talk about the chloroplasts, but they're another ancient bacterium that's trapped that they think was probably what's called a cyanobacteria, or a blue-green algae. These are the ones that when they have the blooms in the lakes here, you're not supposed to go in or let your dog go in. Those are bacterial, cyanobacterial blooms. They call it blue-green algae, but it's actually not an algae. It's not a plant at all. And so there's an ancient trapped photosynthetic bacteria in addition to an ancient mitochondria in plants. They have both. So the nucleus contains most of the DNA. It's the site of DNA replication of the chromosomes. Again, the mitochondrial DNA will be replicated there. It's where the RNA is transcribed, the RNA is processed, and those ribosomes, those factories that build proteins, they're assembled inside the nucleus. Importantly, the process of translation, remember translation, we're going from our mRNA to a new language, proteins, so that's when we're translating into proteins. That occurs in the cytosol. So the DNA is replicated in the nucleus, it's transcribed into RNA in the nucleus, it's processed, that RNA has to leave, the mRNA has to leave the nucleus before it can be translated into protein. And we'll see many of those proteins that are translated then have to be re-imported into the nucleus. And I have a slide for that in a little bit. So here we can see, this is the figure from the textbook showing the nucleus, and we can see how it's continuous or contiguous with the endoplasmic reticulum. Topologically, so the chemical composition is similar to the cytosol. Unlike what we see in many other organelles where it has a unique chemical environment, this is the same as the cytosol. There's a note here, and we're not going to discuss it more, but inside the nucleus there's also higher levels of organization. These are not surrounded by membranes, but there's other regions where we have concentrations of certain proteins or certain types of DNA, and those are all present and in a highly organized fashion within the nucleus. But again, not separated out by membranes. Yes?
Do you think you could explain a bit more how the nucleus is topologically equivalent to the cytosol?
We're going to get to that. Yep. So we'll talk about why it's chemically similar to the cytosol soon. So we talked about that freeze-fracture technique that we can use to look at the inside of membranes. We can also use it to look inside cells, which is what we're utilizing here. So the cells are frozen and split, and then they pop apart, and it's like pulling apart an avocado. You know, you'll have one side that'll have the divot, and then the other side has the seed, which is, we'll consider that's like the nucleus. So that's exactly what we've done here. And we're looking at the seed half. What you do is you then spray it out with gold, and you put it through a scanning electron microscope, and you can see the surface of the nucleus. So pretty cool. And what we see is it's peppered with these holes. And there's maybe up to around 400 of these. And these pores are a very unique type of transporter. They're different than all the other ones we've talked about. They're their very own category. And each of those pores can move up to 500 macromolecules per second. So per second, they can move 500 macromolecules. They can move very large molecules intact. So things like ribosomes that are around 30 nanometers across, so pretty big. You can move them in and out. Okay? And the other important thing is that you can be moving things in and out simultaneously. So these are really remarkable structures. And we're going to spend quite a bit of time on those in a couple of minutes. First, we're going to move inside and talk a little bit about the DNA. So the thing about the DNA and the amount of DNA we have is it needs to be compacted in order to fit it into the nucleus. Okay? So we have a large genome, and to get it all in there, we have to fold it up, basically. Wad it up into a more compact form. And what happens is we do this by actually, it's counterintuitive, we make it more compact by associating it with a lot of proteins. And the proteins help to compact it. And so when you have your DNA and your protein together, we call that chromatin. Right? So it's the combination of the DNA and the protein associated with it. And you can stretch these out and you see you can see this form which is called beads on a string, where some of you have maybe seen this before or talked about it, where you have this core cluster of proteins and the DNA is wrapped around those proteins, and then it gets successively more and more and more compacted. So the first layer we have here are these what's called the nucleosome, where we wrap the DNA around this cluster of 8 proteins that are called histones. And so we wrap it around, and then there's a little linker. So this DNA is all one piece, so we wrap it around, there's a little link, we wrap it around again, we wrap it around another one, and that's why this looks like beads on a string. In the next step, what happens is we further compact that. So we take those nucleosomes and we fold them up into what's called the chromatin fiber, which then gets looped, and then the loops get basically wound around each other. And so what you're looking at is you're getting this compaction all the way down. And this then allows the DNA to fit into the nucleus. Okay. So it's an organized, hierarchical folding, starting with the nucleosomes, the beads on a string, and then getting more and more and more compacted. And you'll look more at the structure of that in 230. I just want you to know that it gets compacted. Not only because it then fits into the nucleus, but this poses a problem for the DNA. And the problem is that if we want to replicate the DNA, or we want to transcribe it, remember when we talked about gene expression, and we have to have all those factors coming in and binding to the promoter, right? They can't do that if it's all balled up. Okay, so it has to be decompacted in order for proteins to access it. And once the proteins access it, we can get replication or transcription or whatever it may be. And so what this means is that how we package DNA can regulate gene expression. So there's 2 types of chromatin. We have the heterochromatin, which is tightly packed. Oops. What's going on here? Oops. Geez. Okay. We have our heterochromatin, which is tightly packed, so it's very hard. It's still our bDNA, but we've twisted it all up and associated with proteins, so it's very hard for anything to get into those major and minor grooves. And then we have euchromatin, which is loosely packed. And so those are regions that are accessible. And what this means is that we talked about gene expression, okay? And the idea there is the idea behind genetics, right? So you inherit the DNA encodes the genes, and if you have a problem in the sequence of your DNA, say in a promoter region, or in the protein coding region, that's heritable and all the way through, and we can trace those through lineages. And I think most of you are familiar with genetics in some form. How many people looked at like the pea plants at some point in their lives? Pretty much all of you, right? So we call that Mendelian genetics, but there's other types of genetics that are all about this inheritance with the DNA. When we're looking at epigenetics, we're looking above the sequence of the DNA. So what these traits are, is that if you were just following the trait, this person has, I don't know, like they have a blue moon on the back of their hand or something, I don't know. And if you trace that trait all the way through a family tree, it would look like it's an inherited trait. But if you compared someone with the blue moon on the back of their hand to someone that didn't have it, you wouldn't find any differences in the genes. And so what we're looking at with epigenetics is it turns out that how you can express genes can also be inherited. Okay? So I'm going to give you what I think is the most straightforward and hilarious example of epigenetics. And so this is this study here, transmission of stress-induced learning impairment and associated brain gene expression from parents to offspring in chickens. Doesn't sound like a very fun study to read until you hear what it is. So what they did in this study was they took 2 chicken coops. Okay? The chickens were, I don't know if they were all genetically identical. I can't remember how it was set up. It's not hugely important, but what they did was they took 2 chickens and they raised them, and the chickens were happy and they would be able to find food. And you know, if you gave them food, they would find it, and they, you know, lived normal chicken lives. What they subsequently did was they took one of those chicken coops and they exposed them to heavy metal music and flashing lights for X number of hours a day, meaning they're somehow disturbing these chickens, making these awesome heavy metal chickens. And so they did this to these chickens. And what they found was that those chickens in this disturbed environment now couldn't feed properly. They had trouble finding their food sources. Okay? Take all these chickens, breed heavy metal chickens and not heavy metal chickens. Take the eggs, the chicks, put them both into a nice, quiet chicken house, chicken coop. The calm ones, the offspring, no problems. The heavy metal chickens, even though those chicks have never been exposed to flashing lights or loud music, can't find the food. In the same environment as the calm chickens. The offspring of those chickens, again, can't find the food. So they could trace this down 2 generations. That looks genetic, but it's not. It's strictly environmental. And what was happening was that those stressors were modifying the way the DNA is packaged, or there's other covalent modifications that can occur in the DNA. But what they're doing is they weren't changing the sequence of the DNA. So they weren't changing the genetics, but it was altering how the genes were expressed. Okay? And so, of course, we're really interested in epigenetics because we thought we understood genetics pretty well. And we know about lots of genetic diseases. Okay? And so starting with I think sickle cell anemia was the first genetic disease identified. We know lots about genetic diseases, but it turns out that many diseases probably also have an epigenetic component, especially cancers. And so what this means, the terrifying thing about this, is that what your predecessors have done, your parents, your grandparents, your great-grandparents, the environmental exposures they had in their lives actually affects your gene expression.
How would it work in the cancer example? How would it work if, how do environmental factors from somebody else's generation affect the potential of?
Same as with the chickens. So you change the environment of those chickens, and it's passed on. How the DNA is packaged is passed on to future generations.
And how would that lead to cancer being more?
Oh, so how does that lead to cancer? So the way cancer works is that it's a combination of shutting some genes off and other genes on. And that can happen genetically, so some genes can be more prone to being on. So things like oncogenes, you pick up a mutation that makes them more likely to be on, that can contribute. But you can also epigenetically receive that trait so that they're more often on. We'll get back to the cancer in, I think that's the last one we do. Yes?
Out of curiosity, can epigenetics, is that how they're determining that, like, for example, if your parents had mental health issues, it's going to be passed on?
Yeah, so does it relate to mental health issues? It's very possible. I don't know much about those studies. I'm sure if you did a quick sort of PubMed search, you would probably be able to find lots. I would be really unsurprised I would be very surprised if there was not an epigenetic component to many diseases. And of course, you know, there's a huge range of different diseases there as well. Yeah. Yes?
Does this apply to all organisms? Like, will we see the same reaction from humans, or does it apply in like different varying degrees?
Oh, so, yeah, so we basically more or less would see it in all eukaryotes. There's versions of it in prokaryotes that the way it actually works is different. They have kind of a similar thing, but it's prevalent in all eukaryotes. And so who remembers our friend Lamarck? Does anyone remember, what was Lamarck's kind of famous thingy about giraffes?
Oh, yeah, he said that.
So, yeah, what did he say about the giraffes?
That basically their necks were like stretching.
Yeah, so his theory was that they had short necks, but the leaves were up, so they kept reaching, and then they got longer and longer and longer necks, right? And we all laughed. What a fool, Lamarck. Lamarck, epigenetics. That's exactly what this is. The environment influencing gene expression. So basically, turns out, Lamarck was at least partially right. Okay, so here we have our nuclear envelope, and it's surrounded by our double membrane, okay, that goes on to become the endoplasmic reticulum. Just a reminder of how this arose. And now we're going to look at the heart of this. This is what we call the nuclear pore complex. So the nuclear pore complex, and the reason that the cytosol and the nucleus have the same chemical environment, this is what we call a gated transport. Gated? And what that means is that small molecules under 5 kilodaltons just move through. They move in and out. There's no regulation. It's completely passive transport. Simple diffusion. Well, I guess it would be facilitated diffusion. Sorry, it's facilitated diffusion, not simple diffusion. So small things can just pop in and out of here. Okay, sugars, building blocks, things like that, moving in and out. However, for larger molecules, they're moved via active transport. And so that means that you can direct whether they're moving in or out at any given time. And so this is what makes nuclear transport so unique. It's a combination of facilitated diffusion and active transport with selected molecules. If we look inside the nucleus, so in the top left, we can see transcription occurring and 3 types of RNA. There's more than these 3, but the 3 that we often talk about are rRNA, ribosomal RNA, which is used to make the ribosomes. mRNA, this will be our template for proteins, and tRNA, which is utilized in the process of translation. So the processes of transcription, of course, have to be mediated by proteins, the RNA polymerase, and there's a whole bunch of other proteins. Proteins are actually made, though, in the cytosol. So what this means is that our RNA, all of this RNA, the ribosomal RNA, mRNA, and tRNA, are all required to make proteins. They get made in the nucleus, and all of them have to be transported out into the cytosol. And in the cytosol, we can make proteins. And those proteins, in turn, you can see with the purple arrows, some of them return back into the nucleus where they're required for things like replication, chromatin assembly, processing the RNA, and so on. So what this means is we have to really specifically move some things in to the nucleus, and we have to be able to specifically move other things out. And so the process we can see here, and what I'm going to start with, or maybe end with today, is we're going to start, it's controlled by a very important class of switch protein, and these are called monomeric GTPases. Okay. monomeric GTPase. So we talked about ATPases in the last class, and we remember that those are enzymes that speed up the hydrolysis of ATP to ADP and orthophosphate. So a GTPase, GTP, water, GDP plus free inorganic phosphate. So the way they work is that we start out with our GTPase, and it can be in two forms. When it's bound to GTP, it's on. When it's bound to GDP, it's off. And they're actually structurally distinct, and we know when we change the structure of a protein, we change its function. How we get from GTP to GDP is in the name. Water comes in, okay, and our orthophosphate leaves. And the GDP stays stuck. Okay. To turn it back on, what we do is we kick the GDP out. This is very important. GDP leaves. And that empties the binding spot, and a brand new GTP comes in. Okay? The GDP does not get this doesn't happen. It doesn't get phosphorylated back to GTP. And I always ask that on exams, and students always get it wrong. Not all students, some. So the GDP is not phosphorylated back to GTP while it's bound to the GTPase. It actually has to get kicked out. It'll be rephosphorylated in another process by another enzyme, but not here. And so this is the key, the way these switches work. And I'm going to start with these switches in the next class, but we're going to leave it there, and we'll see everybody on Friday.